Thank you so much, Dr. Minzuk. So in the interest of time, we'll move forward with Dr. Allison Hanaford, who will be presenting on the immunological origins of Leigh syndrome.
Sorry for a few. Is this visible in presenter view for everyone or?
Yes, it looks great, Allison.
Oh, okay. Great. Good morning, everyone. I'm Allison Hanaford. I'm a postdoctoral research fellow at Seattle Children's Research Institute. And today I'm going to talk about my work on the immune system and Leigh syndrome. Our lab primarily uses the endospore knockout mouse model of Leigh syndrome for our studies. And this is a mouse model that closely resembles human Leigh syndrome. The mice have a mutation in the endospore gene, which has also been shown to cause Leigh syndrome in human patients as well. Like most human Leigh syndrome patients, the mice are born healthy, and then they start to develop disease that progresses very quickly. And they have symptoms like ataxia, weakness, seizures, muscle wasting. And importantly, they develop the characteristic symmetric bilateral lesions in the brain. Here's an image of a mouse brain from one of our endospore knockout mice that has Leigh syndrome. And we can see the red arrow is indicating the site of a lesion by that sort of bright blue area. And if we zoom into this area here, what we see in this lesion is that the cells are highly positive for a marker called IBO1, that's the magenta color, and that stains immune cells, particularly innate immune cells called macrophages. So we can see that these lesions in Leigh syndrome are very enriched in different types of immune cells. So my lab has been asking, is Leigh syndrome an immune-mediated disease? And does inhibiting the immune system actually prevent Leigh syndrome in our endospore knockout mice? So one of our initial experiments exploring this question was we started treating these mice with a drug called pexidartinib at weaning age, and that's 21 days old for mice. Pexidartinib is a drug that inhibits a marker, a protein called CSF1R. CSF1R is expressed on the cell surface for a variety of types of macrophages. And when you inhibit this signaling protein, that eliminates the innate immune cell macrophage population. And we found that treating endospore knockout mice with pexidartinib was extremely effective. So what we can see in the image on the left, this is an image of from the brain of a mouse that was not treated with pexidartinib, and you can see that very red lesion within the dotted line that's enriched with all those immune cells. And on the right, we see there's nothing there. That is an endospore knockout mouse that had been treated with pexidartinib its whole life. And if you look at the survival, the gray line is untreated mice and green is mice treated with pexidartinib. And importantly, these mice, they aren't actually dying of Leigh syndrome. Unfortunately, what ultimately leads to death is toxicity due to the high dose of the drug. So after these results, we became interested in what specific types of immune cells are involved in the Leigh syndrome disease process. And in particular, we were interested in macrophages. And macrophages are a type of innate immune cell that are found throughout the body. They are characterized by the ability to perform phagocytosis, which means they engulf, eat, and ingest pathogens. They're essentially patrolling for pathogens and can recruit other immune cells. Most tissue types throughout the body have an innate population of macrophages that is specific to that tissue type. In the brain, these are called microglia. And there are also macrophages that are circulating in the blood. As I said, microglia are the macrophage population in the brain, and they're the primary type of immune cell we find in the brain. They respond to infection And they also clean up debris from damaged cells, eliminate dead cells. They're sort of the brain's housekeepers, and they prompt repair after infection. This is an image, a close-up image of microglia in the brain. The image on the right is a blown up version of the white box in the image on the left. And what you see here, microglia stain positive for both IBA1, which is magenta, and another microglia specific marker called P2RY12, which is in cyan. And you can see that these cells, they have a pink cell body and these spindly processes, which is how they're sensing their environment there. We can see that the lesions of Leigh syndrome, we're looking at the lesion from an endospore knockout brain here, are highly enriched in microglia, which are cells that are positive for both IBA1 and P2RY12, or pink and cyan. So this means this is a highly inflamed area, and persistent neuroinflammation can lead to cell death and neurodegeneration. And people have been interested in microglia in Leigh syndrome for quite some time, actually since the very first description of Leigh syndrome was published in 1951. But what's also interesting is that in this description, Leigh used a very non-specific stain called a Nissl stain. So it's staining not specifically for microglia. And actually, IBA1 is not a specific stain either. So we often are frequently describing these as microgliotic lesions, but there could be other immune cell types there. And we also wanted to know, how important are microglia to driving the disease? So we used another mouse model to address this question, a special mouse model called fire mice. These mice have a deletion in the CSF1R gene, and mice that are homozygous for this particular mutation do not have any microglia or tissue macrophages. Their circulating macrophages, however, are intact. So we bred our fire mice without microglia with our endospore knockout mice to generate endospore knockout mice that do not have any microglia. And we found that eliminating the microglia does extend the survival of the mice a little bit. We see that in the red line. That is the endospore knockout mice that do not have microglia. But ultimately, it doesn't actually prevent disease. It delays the onset slightly. And the endospore knockout mice without microglia still develop lesions. The image on the left is an endospore knockout mice with intact microglia, and the one on the right is from an animal an endospore knockout mouse that does not have microglia. And you could see these two images look different, and we could see this IBA1 staining cell population over here. And actually, this cell population can't be microglia because these mice don't have microglia. So there's some other cell type present here. And as I mentioned, we've been using a non-specific stain, IBA1, like Leigh, similar to the non-specific Nissl stain that Leigh originally used. So there's clearly some other cell type that is present in these lesions. We next decided to look at circulating macrophages, which are macrophages that are patrolling in the blood, and they then migrate from the blood to a site of inflammation in response to inflammatory signals. Usually these aren't found in the brain, or if they are, they're in relatively small numbers. And we used an additional marker called CD45, which is seen here in yellow. So magenta is still IBA1, and circulating macrophages will be positive for both IBA1 and CD45. And here we see a few scattered circulating macrophages in the brain of this mouse. And the image on the right is a blown up version of the white box on the left. And what we found is that lesions in endospore knockout mice are highly enriched in circulating macrophages. So these circulating macrophages are positive for both CD45 and IBA1, so both yellow and pink. And in the case of immunofluorescence, that overlap can look white in some places. And this is a very interesting finding that we recently published in brain pathology. And we also found that the Leigh syndrome mice that have without microglia their lesions are also full of circulating macrophages. We could see in the image on the right, those cells there are IgG1 positive and CD45 positive as well. So the lesions are forming in the mice without microglia because of these circulating macrophages. So this cell circulating macrophage population contributes to the pathology of Leigh syndrome. And we've also been asking, beyond innate immune cells, what other immune factors contribute to endospore knockout disease pathology? We examined the brain stems of endospore knockout mice, and we chose the brainstem because that is where these lesions formed. And we analyzed the expression of different cytokines, and cytokines are signaling molecules, specifically inflammatory cytokines. We found elevated levels of a cytokine called interferon gamma. We can see that as the bar on the right, is the sample from the knockout mouse. And interferon gamma is a really important signaling molecule in the immune system. It supports many immune pathways. It activates other immune cells. And interestingly, it's extremely highly expressed in many autoimmune diseases. So to investigate this, we took a similar approach where we bred our endospore knockout mice with mice that do not express interferon gamma. So, interferon gamma knockout mice. And this generated Leigh syndrome mice that do not have any interferon gamma. And here we found that not expressing interferon gamma is beneficial to endospore knockout mice, that endospore knockout mice that express interferon gamma are the blue line. Those without interferon gamma are the pink line. So you see a nice survival benefit. And interestingly, if you have one copy of the interferon gamma gene, you have a somewhat in the middle survival. So there's a dose-dependent effect. The less IFNG you have, the better you'll survive. So we found that microglia and circulating macrophages are both contributing to Leigh syndrome pathology, and elimination of microglia alone is not sufficient to prevent Leigh syndrome. And IFNG signaling is also important to Leigh syndrome pathology. And we believe that the immune system's a very promising therapeutic target for treating Leigh syndrome. And we look at mitochondrial disease based on this pathway, where mitochondrial dysfunction is leading to cell stress, which through some as yet to be identified mechanism leads to activation of the immune system, which leads to inflammation, lesions, and neurological disease. And this mechanism for mitochondrial disease development presents several areas where we can interfere pharmacologically to treat patients, either by directly targeting immune activation or potentially the mechanism that actually activates the immune system. And to end, I would like to acknowledge my mentors, Simon Johnson and Phil Morgan, and also my lab mates here, and in particular, the Northwest Mitochondrial Research Guild, which has funded this project.